everyone. I'm Meredith Ashby, Director of Market Strategy for Microbial Genomics here at PacBio, and I want to welcome everyone to the PacBio webinar series, with today's topic focused on how PacBio hi-fi sequencing could provide you with richer information about your microbiome samples, featuring our guest speaker, Jeanette Gehrig from Siolta Therapeutics. First, some housekeeping. The presentation portion of the webinar will be followed by a Q&A session. You're welcome to submit your questions at any point during the webinar by typing them into the question tab on your dashboard. We will be recording the webinar and making it available for download in the next few days. I also want to point out that we have provided you with some useful sequencing handouts that you can download now from your attendee dashboard as well. So before I introduce Jeanette, I'd like to begin with a short overview of PacBio hi-fi metagenomics for those of you who may not already be familiar with our technology. So PacBio hi-fi reads are unique in that they combine both long read lengths with high single molecule accuracy. And the way we do that is by sequencing multiple passes over the same individual molecule, which allows us to generate a very high consensus accuracy hi-fi read. As you can see on the graph to the right, the reads fall in a very tight distribution of accuracy and are over 99.9% accurate. So what does that mean for metagenomics? So for metagenomics, that means our 16S sequencing is high accuracy single molecule, and we can provide up to 3.6 million of these hi-fi reads that incorporate full 16S or larger sequences. For shotgun sequencing, we can provide up to 2.4 million reads using 10 KB libraries on our Sequel II and 2E systems. For 16S, we have recently updated our PacBio protocol so that you can now sequence up to 192 samples in a single SMRT Cell 8M. And one of the appeals of this approach is that it's inexpensive to start up. You only need to order 28 barcoded primers to do a full 192 plex run, and that costs you only $8.50 a sample at 192 plex, and that refers to our North American pricing, and it includes only the reagent cost. So if you do this at a service provider, there will be a service markup. Another option that's very popular among our customers, and which Jeanette is going to be talking about today, is Shoreline Biome. So Shoreline Biome makes a terrific strain ID kit that produces a longer amplicon that covers the 16S and highly variable ITS domain and anchors in the 23S region. When you combine this with PacBio sequencing and their SB Analyzer software and curated full-length 16S database, you can easily do ASV identification and track trains across different samples and from experiment to experiment as well. For shotgun sequencing, one of the advantages of hi-fi is that our data is very rich in functional information. So most reads yield multiple annotation and contain multiple genes. So on the left, you can see that on average, a single hi-fi read, since it is so very long, contains about 8 complete genes. And so one of the key advantages of this is, as you know, in microbes, genes that are involved in the same functional pathway tend to be grouped together in the genome as well and form an operon. And so it's frequently you can recover entire operons, which helps with functional annotation. On the right, you can see that there's a tremendous number of predicted genes per sample, right? So here we have a range of different human microbiome fecal samples, and you can see that in each one of these samples, the total number of predicted genes is enormous, on the order of 15 to 20 million. And if you dereplicate those to just the unique reads, it's still a very impressive number, and frequently more than people would expect, given the relatively fewer number of reads, because those reads are so long and such high quality. And Jeanette is going to touch on this as well in her presentation. Finally, hi-fi reads produce highly contiguous novel references from unculturable species. So hi-fi accuracy allows reads from closely related species to be distinguished. So frequently a species is defined as an organism which is at least 3% different from another. And the fact that our accuracy is higher than that allows us to distinguish reads and assemble even species that are very closely related or different strains into highly contiguous contigs and sometimes even closed genomes. So high-quality mags can be assembled from only 15-fold hi-fi coverage, since there's no error correction step that needs to be built in. And that, in addition, gives you access to lower abundance species. So it's now my pleasure to introduce our featured speaker, Jeanette Gehrig. Jeanette is a senior scientist at Siolta Therapeutics. She first began studying the interplay between the gut microbiome and early life nutrition on development during her doctoral studies in Jeff Gordon's lab at Washington University. There, she gained extensive experience analyzing and integrating large, diverse data sets, including metagenomics, metabolomics, and proteomics data from children and infants, as she sought to understand how the gut microbiome impacts childhood undernutrition. More recently at Siolta, she is developing and testing preclinical microbiota-directed therapies for the treatment and prevention of allergic disease and other indications. Siolta's lead therapeutic was evaluated in a first-in-human safety trial in 2019. In her talk today, she will share results from a collaboration between PacBio, Siolta Therapeutics, and Shoreline Biome, using samples from that trial to explore what additional information can be gained by reanalyzing samples from that trial using hi-fi sequencing. Welcome, Jeanette.
Thank you, Meredith, for that introduction. And let me share my screen here. All right, and thanks again, Meredith. And I'm excited to share with you all a comparison of two different sequencing methods, so long read and short read sequencing, for microbiome profiling. To put this analysis in context, I want to introduce the company that I work for. So Siolta Therapeutics is a clinical stage biotech company targeting the gut microbiota to prevent and treat chronic disease. Our lead candidate is a live biotherapeutic product consisting of a defined microbial consortium for the treatment and prevention of allergic disease, including atopic dermatitis, food allergy, and allergic asthma. As part of Siolta's clinical development pipeline, we integrate microbiome data with clinical data, as well as metabolomic data and immune data. We often find ourselves asking, how can we best identify gut microbes as potential therapeutic targets? How can we determine the pharmacokinetics and pharmacodynamics of a live biotherapeutic product, or LBP? For example, how long an active ingredient strain remains in the gut. How can we better understand an LBP's mechanism of action? And how can we target at-risk individuals for a particular microbiome intervention? The answer to all these questions involves microbiota or microbiome profiling. So this is essentially which bacteria or which taxa make up the gut microbiota and what their potential functions are. To put microbiota profiling in context and understand the current landscape, I'm going to go through a brief history of methods used for microbiota and microbiome profiling. So in the early 20th century, microbiologists first noted that for complex microbial samples, there was a discrepancy between the number of organisms that could be viewed through a microscope compared to the number of organisms that could be cultured on a plate. This became known as the Great Plate Count Anomaly. So there were a far greater number of organisms that could be viewed in a microscope compared to cultured. And this led to a dependence on culture-free methods for microbiome profiling. In the 1970s and 80s, scientists pioneered the use of 16S ribosomal rRNA comparisons to reconstruct phylogenies of organisms. And this, in combination with new technologies such as PCR and Sanger sequencing, led to a dominance of 16S ribosomal profiling for microbiome profiling. And the highly conserved 16S region contains multiple variable regions interspersed with conserved regions. With the advent of next generation sequencing, so massively parallel, inexpensive sequencing, the predominance of 16S ribosomal rRNA profiling continued because these short variable regions could be primed and sequenced. And typically select variable regions were chosen because with next generation sequencing, the read length was limited. With the continuation of next generation sequencing and the continuous dropping in costs of next generation sequencing, it also became possible to go beyond amplicon sequencing and to sequence all the DNA in a particular microbial sample. And this is known as whole genome sequencing or metagenomic sequencing. And this allows, in addition to understanding the taxonomy or which organisms are in a sample, it also gives information on the functional ability of a particular microbiome. But one of the downsides of short read sequencing is that with short reads, it's difficult to construct genes and genomes. Recently, improvements in third generation or long read sequencing have led to a decrease in the error rates for long read sequencing. For example, the improvement of circular consensus sequencing. So now with these newer third generation sequencing methods, it's important to compare them to the more established short read methods for microbiome profiling. For this methods comparison, we focused on 4 different sequencing methods. So for amplicon or 16S sequencing, we used a short read method with Illumina and also a long read method. And similarly with the whole genome or metagenomic sequencing, we compared short read metagenomics to long read metagenomics. The purpose of amplicon or 16S bacterial or 16S sequencing is limited to bacterial taxonomic profiling. Whereas with metagenomics, both short read and long read, you can go beyond just bacteria and look at other non-bacterial members of the microbiota. And also you can get information because you're sequencing all the DNA in a sample, you can get information on the function of the microbiome, as well as recovering genomes from complex samples. Because this methods comparison will inform Siolta's future approaches for microbiome sequencing for both preclinical and clinical studies, we wanted to focus on relevant samples from a clinical trial. So we chose samples from Siolta's first phase 1B first-in-human safety trial of our lead live biotherapeutic product, or LBP. About 60 participants participated in this trial, and they were treated with either our LBP or placebo twice daily for 4 weeks. We selected a subset of 6 participants from this larger trial and looked at 2 fecal samples from each of the 6 participants. 1 sample from baseline before treatment, and then 1 sample after about 4 weeks of treatment with the live biotherapeutic. We were interested in comparing how long-read microbiota and microbiome profiling methods compared to traditional 16S and metagenomic sequencing. So we looked at the pipelines and outputs, and we also looked at the utility of the data, including taxonomic resolution, functional profiling, detection of active ingredient strains, and as well as microbial gene recovery. So as I mentioned, we focused on these 4 different sequencing methods. So it's essentially easy to view it in a matrix. So for amplicon sequencing, we looked at both V3, V4 short read amplicon sequencing, as well as long read amplicon sequencing. And then for the whole genome sequencing or metagenomic sequencing, we performed both short read metagenomic sequencing and long read metagenomic sequencing. For the amplicon sequencing comparison, for the short reads, we focused on an amplicon that encompasses the variable regions 3 and 4, which is about a 460 base pair amplicon. And this V3, V4 16S sequencing is part of Siolta's current pipeline, and it's also frequently used in the field. For example, it was used with the Human Microbiome Project to profile diverse samples, diverse microbial samples. For the long read 16S amplicon counterpart, we turned to this amplicon that includes the entire 16S gene, in addition to the internal transcribed spacer region and part of the 23S gene. And the company Shoreline Biome provides a kit called Strain ID that allows you to prepare these long 2.5 kb amplicons for sequencing with the PacBio Sequel II. And then with the, so overall, if we look at the data output per sample, data output is comparable between both amplicon sequencing methods and also between both whole genome sequencing methods. So you can see with the whole genome sequencing or metagenomic sequencing, we have about 100 times the data that we do for the amplicon sequencing. So because these different sequencing methods have different requirements for sequencing as well as different data outputs, we use different library prep methods as well as different software for data analysis for these 4 different methods. For example, the cationic detergent CTAB can be used as part of the DNA extraction protocol for short read sequencing. But for long read sequencing methods, we need a more gentle DNA extraction. So we use the QIAGEN AllPrep Power Fecal Kit. To see if DNA extraction method could impact the microbial profiles downstream, we wanted to compare DNA extraction method. And so we did this with the strain ID sequencing. And so for the strain ID analysis, we included multiple replicates from each of the 12 fecal samples, and each replicate had its DNA extracted with a different DNA extraction method. Our main questions for this analysis focused on 4 different areas: taxonomic profiling, functional profiling, detection of an LBP active ingredient strain, and metagenomics assembly and binning. For taxonomic profiling, we first wanted to see if DNA extraction, the method of DNA extraction, could affect microbiota profiles. We wanted to compare these overall taxonomic profiles and determine how many unique taxa were detected with each method. We were also interested in looking at the taxonomic resolution with each method, so looking at lower levels of taxonomic resolution, such as species and strain. And we wanted to see if there were similar microbiota taxonomic changes across participants from baseline to treatment, as well as across methods. So first, we wanted to see if different DNA extraction methods could impact overall microbial profiles. So for this, we focused on the strain ID analysis, where we had multiple replicates from each sample that had DNA extracted with different DNA extraction methods. So on the left here, I'm showing a PCOA plot of the beta diversity metric Bray-Curtis, and each dot represents a replicate. And I've colored each replicate by the participant from which it came. So you can see the replicates tend to cluster by participant, and this clustering by participant is significant. However, if we look at the DNA extraction method of a particular replicate, so in this graph on the right, I'm coloring samples by the DNA extraction method. You can see that there's no significant clustering by DNA extraction method. We still see that clustering by the participant. So this is telling us that DNA extraction method does not have a drastic impact on overall taxonomic profiles. To compare taxonomic profiles across methods, we wanted to look at higher levels of taxonomy. So here I'm showing the phylum. So we combined all the samples together and then looked to see which phyla the majority of sequences was assigned to. So overall, we see across all 4 methods, V3, V4, strain ID for amplicon sequencing, and short read and long read metagenomic sequencing, we see a predominance of firmicutes for across all 4 methods. Even though we see broad similarities, so the predominance of firmicutes, as well as we see a lot of Bacteroidetes and actinobacteria, we do still see some differences across these 4 methods. So, for example, we see with strain ID, there's a greater proportion of unknown phyla in pink here. And also with short read metagenomic sequencing, we see a greater proportion of actinobacteria compared to the other methods. Both of the short read methods show a greater relative abundance of the phyla Verochromicrobria. So this is shown in gray. We see greater relative abundance of this particular phylum with both short read methods. And we also see with the 2 short read methods the presence of the Archaea phylum Euryarchaeata, and this is in purple up here. And we don't see this with both of the long read sequencing methods. We can also look at a slightly lower level of taxonomy. So here again, I'm showing the combination of all samples profiles at the family level. And once again, we can see some overall similarities and differences across the 4 methods. So for example, across 3 of the methods, we see a predominance of the family Lachnospiraceae, with the exception of the strain ID sequencing. And with strain ID, once again, we see a greater proportion of unknown families in this case. So this could potentially make up for some of the differences we see across the 4 methods. We also see pretty high abundance of Ruminococcaceae in blue here, as well as Bifidobacteriaceae and Bacteroideaceae in yellow at the top. We see that both of the amplicon methods have higher representation of the family Erysipelotrichaceae, and this is in green here. And then strain ID has a greater representation of the family Peptostreptococcaceae in pink here. And so these differences could be due to differences in library prep. So for example, biases in amplification, and they could also be due to differences in classification, in taxonomic classification across methods. In addition to looking at overall taxonomic profiles at higher taxonomic levels, we also wanted to compare the relationship between samples within each method at the lowest available taxonomic resolution. So this was typically species or strain level resolution. So within each method, we calculated the Bray-Curtis distances between all of the samples. And I'm showing that here on a PCOA plot. And you can see across all 4 methods, we see clustering by participant. So the dots here are each sample, and they're colored by the participant from which they came from. So for example, we see participant 4 clustering together across all methods. Similarly, we see the same samples from the same participant clustering together. We can perform Mantell tests, so we can see if the distance matrices across all 4 methods are correlated with each other. And we do in fact find that each distance matrix has a significant positive correlation with the other 3 matrices from different profiling methods. So this is telling us that across all 4 methods, the relationship between samples is preserved. In addition to beta diversity, or the relationship between samples, we also wanted to look at alpha diversity across methods. So this is the within sample diversity. And in this case, we're looking at the number of unique taxa detected. So for across all 4 methods, we see here I'm showing the number of unique taxa detected with the 4 different methods. So V3, V4, strain ID, and both short and long read metagenomic sequencing. We see with strain ID, we have the highest number of unique taxa detected per sample. And both of the metagenomic sequencing methods showed a lower number of unique taxa detected. And I'll just mention here that for the long read metagenomic sequencing, we had an issue with participant 6 treatment sample, so we don't have a comparison for that sample. We were also interested in looking at the taxonomic resolution across these 4 methods. So how can we see, how well can we see the lower levels of taxonomy, such as species and strain? So for this analysis, I looked, for each sample, I looked to see the proportion of reads that mapped to family, genus, species, and strain level. And here I'm just comparing the amplicon sequencing methods because most of the taxonomy from the metagenomic sequencing methods are assigned to the species level. And so here I'm comparing the V3, V4 16S sequencing with taxonomy assigned from 2 different 16S databases, Green Genes and Silva, to the taxonomy assigned to the strain ID amplicons. So we can see that strain ID had a significantly improved species level resolution compared to Silva, but not compared to Green Genes. So you can see in green here, comparing the fraction of reads assigned to species, this is comparable between Green Genes and strain ID, but is lower for Silva. And this highlights the impact that a reference database can have on your analysis. Also, it's important to note that V3, V4 taxonomic assignments inherently have a greater degree of uncertainty because we're mapping a shorter region compared to the full 2.5 kb strain ID amplicon. And importantly, only strain ID is able to assign strain level taxonomy. So there's an average across all samples of about 13.4 reads per sample that are assigned to the strain level. With taxonomic profiling, we can look within a particular individual and see if particular bacterial taxa increase or decrease in abundance over time. In this bubble plot here, I'm showing the relative abundances of several species assigned by long-read metagenomic sequencing and comparing baseline to treatment in this one participant. And you can see that certain bacterial taxa are increasing in abundance and others are decreasing in abundance. We wanted to see if there were shared taxonomic changes from baseline to treatment across participants and also across methods. So looking at the family level of taxonomy, we found several families that either all increased or decreased in abundance across multiple methods. For example, we saw within the Clostridialis family 13, across 3 different sequencing methods, this particular family increased in abundance from baseline to treatment. However, there was no family that showed similar changes in abundance across all 4 methods. You can see in the best case, we had 3 out of the 4 methods show similar changes. So to summarize this first part of taxonomic profiling, we found that DNA extraction method does not drastically impact microbiota profiles based on our strain ID analysis. We found that overall taxonomic profiles were similar at higher taxonomic levels, but there were some notable differences. And we found that the relationship between samples, or the beta diversity, was conserved across all 4 methods. We found that strain ID showed the highest number of unique taxa detected per sample, and this was about 260 unique taxa per sample. And for taxonomic resolution, we found that strain ID, only strain ID for amplicon sequencing can assign strain level taxonomy. but levels of species and genus were comparable to V3, V4 sequencing. And we did see similar microbiota taxonomic changes across participants from baseline to treatment, but this was not conserved across all methods. Our next subject of focus was functional profiling, and this was for both metagenomic sequencing methods. We were interested in seeing what proportion of reads had a known functional annotation with each method, and we wanted to see if there were similar microbiome functional changes across participants from baseline to treatment, and similarly across all methods. For our functional annotation pipelines, because we have different read lengths, we use different software to annotate functions. So for short reads, we mapped the clean short reads and used human2 to assign functional annotation. And this provides relative abundance tables of gene families and pathways. And for the long read sequencing, we mapped the hi-fi reads to NCBI's protein database. And then we used Megan6 to assign functions. And Megan6 can annotate functions based on several databases. So we were interested in seeing what proportion of our data was actually useful for in terms of assigning function. And so for each sample, we looked at the percentage of metagenomic reads with a known functional annotation. And here I'm comparing the short read sequencing in blue to the long read sequencing in orange. And with the long read sequencing, I'm just using the annotations from one of the functional databases assigned with Megan6. So overall, you can see there's a far greater proportion of reads that are assigned a known function with long read metagenomic sequencing compared to short read metagenomic sequencing. So only about 34% of the short reads were assigned a known function compared to 63% of long reads. And it's important to note that long reads typically have multiple functional hits per read. So in our case, 2 to 4 functional hits per read. Whereas with the short reads typically had 0 or 1 functional hit per read. And this is because short reads aren't long enough to hit multiple genes, considering the average bacterial gene is about 1 kb. And also mapping these longer reads to functional databases gives us greater confidence because we have entire genes and multiple genes contained within a single read. Similar to taxonomic profiling, we can look at the functions within a particular individual's microbiome and see how the representation of those functions change over time. So here I'm showing the analysis from participant 3's samples. So here I'm comparing the representation of different functions from baseline to treatment. And you can see overall the representation of these different functions remains about the same. There are some subtle increases and decreases. So we wanted to see if there were consistent functional changes across participants and also across both short read and long read metagenomic sequencing. So we performed a differential abundance analysis on the functions separately for long read and short read metagenomic sequencing. And in both cases, we found that there were particular functions that increased in representation from baseline to treatment. However, the exact pathways or functions did not overlap between long read and short read sequencing methods. And this could be due to the relatively small number of samples that were included in this analysis and the fact that different databases were used for functional annotation. And also we have to keep in mind that a far smaller proportion of the data was assigned a function with the short read metagenomic sequencing. So to summarize this functional profiling, we found that a far greater proportion of the reads were assigned a known function with long read metagenomic sequencing compared to short read. And we found independently within both short read and long read metagenomic sequencing There were similar microbiome functional changes across participants, but these particular functions were not shared across short read and long read sequencing methods. Because the participants in this study were treated with a live biotherapeutic, we wanted to see if any of the active ingredient strains could be detected in the treated samples across methods. And we wanted to compare our confidence in strain detection. So for both metagenomic sequencing methods, we were able to map reads, both short reads and long reads, to an active ingredient strain of interest that I'm calling strain 1. And so you can see here I'm showing the participant on the x-axis here, colored by whether the sample was a baseline or a treatment sample. And you can see for both metagenomic sequencing methods, across all participants, the amount of reads that are mapping to strain 1 genome is 0 or close to 0 at baseline. And in most participants, this increases with the treatment sample here. And this was independently verified with a quantitative PCR with strain-specific primers. And you can see that with both qPCR and also with the metagenomics mapping, we see participant 5 has low levels of mapping to this strain, both at baseline and at treatment. When we look at the V3, V4 sequencing, in this case, we're looking at mapping to strain 1's ASV. So the V3, V4 ASV is about a little more than 400 base pairs. So we can see the same pattern where there are low levels of mapping to this ASV at baseline, and this increases by treatment. But we weren't sure how confident we could be that this ASV was our strain's ASV, even though it follows the same pattern. So to investigate this further, we wanted to see how confident we could be that we're actually detecting strain 1. We turned to strain ID. And so with our strain ID analysis, in addition to sequencing multiple replicates from each fecal sample, we also included replicates of our particular active ingredient strain isolates. And we know that many bacteria contain multiple and divergent 16S and 23S genes. And we saw that this was true for our active ingredient strains as well. So here I'm showing 3 of our active ingredient strains. And for each strain, we can see multiple strain ID amplicons for these isolates. For our strain 1 isolate, we can see, we were able to see that strain 1's main ASV separated it from 29 other genomes of the same species in NCBI. And if we were to just look at the full-length 16S gene, so the V1 to V9 region, this would be identical to 9 other genomes. And it's actually just a single mutation in the ITS region that distinguishes strain 1 from all other known strains. And we can actually detect this strain 1 ASV in participant 4's treatment sample replicates. So in green here, these are the reads that map to strain 1's ASV from strain 1 isolates. So we're confident this is our strain. And then over here, I'm showing the 4 replicates at the 2 different time points for participant 4. So in blue, you can see we don't see any counts to this particular ASV, but at treatment, we see this increases. So we can confidently detect strain 1's ASV in these treatment samples. And if we were only looking at V3, V4, we wouldn't be able to confidently say that we're detecting our strain because this region is shared with multiple strains of the same species. So if there were a closely related strain already in the microbiota, we wouldn't be able to confidently detect our strain. Mapping the long read metagenomic reads back to our genomes can also increase our confidence that we're detecting our strain. In this one particular sample, participant 4's treatment sample, we mapped the long-read metagenomic reads to strain 1's genome. And this coverage pattern shows a peak and a trough. And this is hallmark of a replicating strain because reads tend to pile up on the bidirectional origin of replication. So this is suggesting that this exogenously applied strain is not just passing through the gut of this participant, but actually growing and replicating. In addition to mapping reads, we can also assemble reads and map contigs to a genome of interest. So in this same sample, we mapped contigs to strain 1's genome, and we can see two very high quality matches. So there were two contigs that had very high quality matches to strain 1's genome. And these matches are close to 1 million and 2 million base pairs. So this really increases our confidence that we're detecting our strain in these samples. So to summarize, we found that we could detect an active ingredient strain across methods, though not in every treatment sample. For example, with strain ID, we weren't able to detect strain 1 in treatment samples where we could with the other methods. And this is likely due to lower sequencing depth for strain ID. Our confidence in strain detection is much higher with strain ID because we know, particularly with strain 1, we know our amplicon is unique compared to other strains of the same species. And also, long-read metagenomics lets us map longer reads, so increases our sensitivity in detecting our strain. The final area we wanted to look into was our ability to recover genomes from these samples. So this is focusing on the metagenomic sequencing. And we wanted to see how metagenomic assemblies compared between short read and long read sequencing. And we also wanted to see how many high quality MAGs we could detect. So MAGs are metagenome assembled genomes. And we were interested in comparing the diversity of these MAGs across methods. So when we looked at the metagenomic assemblies comparing short read sequencing to long read sequencing, we found that the assemblies were much more complete with long read metagenomic sequencing. So the average N50 for short read sequencing was about 7,000 base pairs, whereas the average N50 for long read sequencing was 146,000 base pairs. You can also see this difference in completeness when we look at the largest contig for metagenomic assemblies. So the largest contig from short read sequencing is this represented by this dot here, and it was typically less than 500,000 base pairs, whereas for long read metagenomic sequencing, the longest contig was typically over 4 million base pairs, and this likely represents a complete or a near complete genome. However, when we summed up the length of the assemblies per sample, we saw comparable lengths when we looked at short read and long read assemblies. And we see two exceptions here. And so for the participant 2's baseline sample and participant 6's baseline sample, we see far shorter assemblies for the short read sequencing. And this is likely due to the fact that these two samples had the fewest number of reads when we looked at the short read metagenomic sequencing. But overall, we see that the length of the assemblies are comparable. And this is telling us that we're able to recover about the same amount of unique genome content from each of these samples using either short or long read metagenomic sequencing and assembly. So for with our metagenomic assemblies, we can then group contigs into bins and begin to better understand which particular taxa we can recover from different samples. So for short read sequencing, we performed assemblies using metaspades, and we used HiCanoe for the hi-fi long reads. And then we used MetaBat2 for both sequencing methods to construct the genome bins. And then we filtered genome bins based on quality thresholds. And so it's important to note that For the short read metagenomic sequencing, I had to relax the this filtering threshold. So normally we only allow bins to contain 10 or fewer contigs, but because the short read metagenomic assemblies were so fragmented, we had to allow a greater number of contigs. And so if we ran this pipeline without relaxing this filtering criteria, we wouldn't have any high quality metagenome assembled genomes. So we relaxed this filtering criteria just for the short read metagenomic sequencing and assembly. And so once these bins were filtered, we had our high quality metagenome assembled genomes, or MAGs. So here I'm comparing the genome bins that we got from one particular sample, so the baseline sample from participant 5. And here I'm showing all of the bins from the short read metagenomic assembly and binning and the long read metagenomic assembly and binning. And in the box, these are the high quality metagenome assembled genomes that pass the filtering criteria. So these are the MAGs, these are the genome bins with the lowest level of contamination and the highest level of completeness. And so when we relax the filtering criteria for the short read metagenomic assembly and binning, we get about a comparable number of high quality MAGs compared to long read metagenomic assembly and binning. So we had 21 high quality MAGs with short read assemblies and 26 high quality MAGs with the long read assemblies. So just two of the MAGs were greater than 98% complete with our short read assembly and binning, whereas 18 of these MAGs were greater than 98% complete with the long reads. And then we just had one MAG from the short read metagenomic assembly that had fewer than 20 contigs. And the numbers here are the number of contigs per bin. Whereas with the long read sequencing, all MAGs had fewer than 10 contigs based on the filtering criteria. And we actually had 6 MAGs that were composed of a single contig. So this likely represents near complete or complete genomes, or in some cases, plasmids. So overall, the assemblies and the genome bins and the MAGs were much more complete with the long read metagenomic sequencing. So for this same sample, when we compare the taxonomy that was assigned to all these high quality MAGs, we actually see that there's a high degree of overlap between short read and long read methods. So we see 13 taxa that are shared between the short read and long read methods. And these are mostly bacteria, but we also see an archaea. So both short read and long read metagenomic sequencing were able to generate this high quality MAG of an archaea, so Methanobrevibacter smithii. We also were interested in looking at the diversity of MAGs across all samples. So we combined all of the MAGs together and looked to see which genus each MAG was assigned to. So here I'm showing the 20 genera with the most MAGs recovered from all of our samples, comparing the long read metagenomic sequencing to short read metagenomic sequencing. And overall, we can see that with long read sequencing, we recovered 201 MAGs across all samples, and they belong to 53 unique genera. And then with the short read metagenomic sequencing, we were able to recover 185 total MAGs belonging to 60 different unique genera. We were able to recover most of the MAGs from the genus Ruminococcus. And we can see with the long read sequencing, we got a greater number of MAGs from this genus. We also saw we were able to recover a lot of Faecalibacterium MAGs from that genus. And we had a significantly greater number of MAGs falling into this genus for the long read sequencing compared to the short read sequencing. Whereas with short read metagenomic sequencing, we recovered more MAGs from the genus Agathobacter compared to the long read sequencing method. But overall, we see similar taxonomy and and similar mags were recovered from both long read and short read sequencing methods. So to summarize this last part of the comparison, we found that the long read metagenomic sequencing resulted in significantly more complete assemblies. And when we actually relaxed the filtering criteria for assigning high quality mags, we got about the same number of high quality mags from both samples. And these mags were similar in taxonomy, comparing both the short read metagenomics and the long read metagenomics. And we got a very good diversity of mags coming from 50 to 60 different genera. So just to conclude this sequencing comparison, overall, we found that all methods generated comparable taxonomic profiles. And then different sequencing methods have pros and cons. And I think the sequencing method should really be chosen based on the scientific question. So for example, if you're interested in detecting taxa at very low abundance, the low cost and the high depth and coverage you can get from short read sequencing can provide a glimpse into lower abundance taxa. And also metagenomic sequencing provides information on non-bacterial taxa. Strain ID sequencing really excels at identifying and tracking multiple strains of the same species. And it uses amplicon sequencing, so it's still relatively inexpensive. And then finally, long read metagenomic sequencing really provides more information on functional annotations. So it provides both higher accuracy and also a greater proportion of the data that's assigned a functional annotation. And long read metagenomic sequencing is also really useful in being able to extract complete or nearly complete genomes from complex metagenomic samples. And with that, I'd like to thank everyone for your attention, and I'd be happy to take questions.
Hi, Jeanette. Thank you for the terrific talk. And as you just noted, we will now have some time for Q&A. So for the Q&A, I'd like to invite Dan Portick to join us. Dan is a bioinformatics scientist here at PacBio and our in-house expert on metagenome sequencing. So let's take a look at some of the great questions that have come through. All right. So the first question is, how many reads were produced in the strain ID experiment?
I can actually show you that. All right, here we go. So for the strain ID experiment, we had about 20,000 reads per replicate.
That's terrific. And I think these were done at 96 plex, right? If I recall, 96
right.
samples per run.
Yeah. It was a little fewer, but I think about 70 or so.
Okay. Let's see. We have another question. This one is for Jeanette. I think, how did you do the analysis of alpha diversity to obtain the total number of unique taxa?
So the alpha diversity was essentially just number of unique taxa at the lowest level of taxonomic resolution for each method.
Awesome. Okay, so here's another follow-up question on the topic of 16S. Are we sure we're not confusing strains with species or genera? Ribosome sequencing, even the variable and ITS domains do not change between strains of the same species, and in some case, even between phylogenetically close species. Can you be more specific? For example, can you distinguish subspecies of lactobacillus? So I have an answer. I bet Dan has an answer. Jeanette, do you have an answer?
So I think with it's with really only with the strain ID fingerprint. You know, if we're talking about amplicon sequencing, I think we're really only able to confidently detect a strain if we know what our particular strain's fingerprint is, as far as, you know, number of 16S genes, how similar or different they are. And yeah, we really can't quite do that with the shorter V3, V4 or other shorter amplicon sequencing.
Yeah. So I'm going to channel my Shoreline Biome friends here and say that the ITS sequence actually does vary even within the same species. So many bacteria, most bacteria have multiple copies of the 16S gene, and some of those copies are different one from the other. And the unique assemblage of all of those sequences together constitute the fingerprint that Jeanette was referring to. And so with Shoreline Biome, what you can do is actually track the appearance of that unique multi-16S fingerprint in your sample so that you can in fact do strain level recognition and tracking in your sample. Dan, did you want to add anything? Did I say it right?
Yeah, I think that covers it.
Okay, great. Yeah, and again, I would encourage you, if you have more questions on that, to reach out to the folks at Shoreline Biome. They are always eager to talk to people about how their product works and how innovative it can really be. Okay, let's see. Question, how do you interpret the results where there's not even one taxonomic group that were increased and decreased in all 4 methods?
So I think because we had only 6 participants for this comparison and 12 samples, it's a relatively small N. And then also our intervention was not drastic, as in, these were otherwise healthy individuals. We didn't have any antibiotic treatment or any severe illness or anything. So I think the overall impact on the microbiota was not drastic. So I think if we had maybe more individuals, we might be able to see more subtle differences. But I think this is just not a great effect size and also a small N problem.
Okay. So another follow-up question, and there's kind of 2 questions on the same topic. So it says, when you say functional assignment for short reads, do you mean per read or on assembled mag contigs? And we had the same question about the HiFi data as well. Do we recommend doing functional assignment on individual reads or on the assembled contigs?
So the functional assignment for both the short read and long read was based on individual reads. But I think with there could be some improvement with, especially with short read metagenomic sequencing, if you assemble first. But that's an analysis that's still ongoing.
Awesome. Dan, do you want to maybe talk about the PacBio pipeline that you've implemented that folks can use and how that works?
Sure. So we do have a PacBio specific pipeline on our GitHub in our metagenomics repository. And this will allow you to carry out functional annotation directly on HiFi reads. And we recommend using the reads themselves rather than the assembled contigs because the reads just give you more information. We have multiple complete genes on every single read. So functional annotation is actually really easy to do starting directly with the reads. And there really isn't any reason to assemble beforehand. And just keep in mind that assembly can, especially with short reads, potentially introduce errors. So performing some type of assembly beforehand might introduce additional bias in terms of functional annotation.
Awesome. Here's another question. Did you try using the NCBI 16S curated database?
For the no, I didn't for the amplicon sequencing. I just used the V3 or sorry, the Silva and the green genes V3 V4 reference database. But that's a good point, and that's something that could improve taxonomic resolution, at least for amplicon sequencing.
Dan, have you ever used the NCBI 16S curated database?
I have, but not for any specific purpose except for testing. So I can't really comment on the quality of it.
Fair enough. Okay, we have time for maybe a couple more questions. How do you account for the higher proportion of unknown reads from the strain ID data?
I think this is due to how taxonomy is assigned with the, so Strain ID provides their own software and database for annotating these long amplicons. So they have their software is SB Analyzer, and then they have the Athena database, which contains the 16S, ITS, 23S amplicons. And in order for a particular ASV or a particular amplicon to be confidently called a certain level of taxonomy, it has to both have 97% identity, and also it has to have 97% identity or more to just one particular amplicon. So I think it's a higher threshold to pass, and I think there are a lot of bacteria that we haven't really sequenced and annotated, especially these longer amplicons that this database just hasn't been around as long. So I think I think it's largely the the limitation of the database and also the high stringency for confidently calling a particular level of taxonomy.
Great. And see, let's do one last question. This is a question I think we hear a lot from our customers. Why were functional assignments of short versus long reads compared using different methods? The approaches underlying the Human2 and Megan are very different. Dan,
right.
Do you want to take this one, or Jeanette too, whichever?
Maybe Jeanette, you can start.
I can start, yeah. So I think the methods for assigning function are really tailored to either short reads or long reads. So we sort of wanted to choose the best or the most used, with either short reads or long reads. But that's a really good point, and that is a question I'm looking into. So if we can treat our short reads as long reads, maybe, so first assemble the reads, and then also retain the coverage of those reads as well, and then put that through the long read database, so we can get a comparable analysis.
Awesome. And Dan, maybe you could go into a little bit like, why is it that short read tools don't often work that well with long read data?
Yeah, that's a great question, and it's something that we see a lot. So, for example, with taxonomic annotation, a lot of these short read methods will use very short k-mers to assign reads to particular taxa. And that simply doesn't work with long reads. Because we have so much additional information, we can actually do entire alignments of multiple genes to these databases, whether that's a nucleotide database or a protein database, and we'll end up getting multiple genes on a read assigned to a particular taxon. And that actually makes us much more confident in our species identification with the long reads. And we've noticed if you try a short read method using long reads, for example, using Kraken or some other taxonomic assigner, it'll produce lots and lots of false hits that just simply aren't in the data set. And we can confirm this using our mock data and other things too. So for all intents and purposes, short reads and long reads really do need to be treated as distinct. And so that's why we're working hard to produce pipelines that are specific for our hi-fi data that allow you to do the same things that you would do with short reads and much, much more.
Awesome. Okay, so I think that's all the questions we have time for, but if you have more questions at the end of the webinar, there'll be an opportunity where you can click follow up with the PacBio scientists, and I definitely encourage you to do that. Our scientists are super nice and friendly and not scary at all. Okay, so in closing, thank you to both Dan and Jeanette for sharing your time and your perspective and expertise on hi-fi sequencing for metagenomics. And in closing, I want to briefly mention 3 things. First, we have recorded the webinar, and it'll be available for download in the next few days. And keep an eye out for a follow-up email with a link to the recording. And then second, you can stay up to date with all of our upcoming webinars by visiting and bookmarking pacb.com/events. And finally, immediately following the webinar, you'll receive a very brief questionnaire. Please do take the 2 minutes that is required to fill it out. because it helps us develop new content for future webinars. And thanks again for joining us today. I hope to see you again on a future webinar. And with that, take care and have a wonderful day.